epiglottis is a cartilaginous structure that is present epi or upon glottis. Glottis is nothing but an opening for air to enter inside a respiratory tract. And just above glottis, you can see a leaf-like cartilaginous structure, which is your epiglottis. Structurally, epiglottis contains a core that is made up of elastic cartilage. And this core made up of elastic cartilage is covered by a layer of mucosa. You can see right here. Here's your core made up of elastic cartilage, not your core, but the core of your epiglottis, which is covered by mucosa. Since it has a mucosal covering, it is obviously going to have a mucosal epithelium lining its surface, right? So, in order for you to know the histology of epiglottis, you have to know the lining epithelium of epiglottis, and you can remember that by simply using your common sense. See, this is how. So your epiglottis, it contains two surfaces, an anterior surface and a posterior surface, right? The anterior surface is continuous with the tongue. The posterior surface is continuous with the trachea. And that is why the lining epithelium of the anterior surface is going to be the same as that of the tongue, since it is continuous with the tongue. And the lining epithelium of the lower portion of the posterior surface is going to be the same as that of trachea, since it is continuous with the trachea. Simple logic, right? So the final verdict is that, the final concept is that the majority of the epiglottis is lined by, how much majority means? The anterior surface and the upper portion of the posterior surface. The amount that I have shown in green, I have highlighted in green. It is lined by the same epithelium lining the tongue, and that is NKSS, or non-keratinized stratified squamous epithelium. The lower portion of the posterior surface of the epiglottis is lined by the same epithelium lining the trachea, and that is a respiratory epithelium. Now, big question, what is respiratory epithelium? See, respiratory epithelium is going to be very significant throughout your respiratory system, and it is none other than pseudostratified ciliated columnar epithelium. The significance is that you have these tall columnar cells, right, along with these small basal cells that are arranged in a single layer, but since nucleus are present in two different layers, it seems as if the cells are stratified or in two different layers. However, that is a pseudo or false. The surface, the free surface, is lined by cilia. The significance of cilia is that it beats continuously in a rowing fashion outside or towards the oral side. As a result, if any foreign particle gets inhaled in, it can get easily trapped and beaten out. Okay, so that's the significance of respiratory epithelium. So let's get into the histology. I said that the core of epiglottis is made up of elastic cartilage. And the elastic cartilage core is covered by a layer of mucosa. Mucosa contains the epithelial mucosa and lamina propria. The epithelial mucosa lining the anterior surface of epiglottis and the upper part of the posterior surface of epiglottis is the lingual mucosa, or the same that is found in tongue, that is non-keratinized stratified squamous epithelium. The lining epithelium present on the lower portion of the posterior surface of epiglottis is the same that is found in larynx, or the laryngeal mucosa, that is pseudo-stratified ciliated columnar epithelium. In submucosa, you can find seromucous glands. So these are the slides that could be provided to you in order to identify in your anatomy practicals. What can you see here? You can see a core that is made up of elastic cartilage, and the core is covered by a covering of mucosa. The mucosa has epithelial mucosa and lamina propria. The epithelial mucosa on the anterior surface is non-keratinized stratified squamous epithelium, which is the same epithelium lining your tongue. On the lower part of the posterior surface, the lining epithelium is respiratory epithelium, which is the same epithelium lining your trachea. Within lamina propria, you can see bunch of glands. These are seromucous glands. Up next, we have trachea. So trachea, it is covered by a number of C-shaped hyaline cartilages. We know that from our basic anatomy. Here's the transverse section of trachea. You can see that there's trachea anteriorly, and behind trachea is the food pipe or esophagus. And the trachea contains the C-shaped hyaline cartilage. Now the reason why this hyaline cartilage is C-shaped and not a circle is because if the cartilage were a circle, it would have obstructed the esophagus. That is why posteriorly we have free ends of this C-shaped hyaline cartilage. We have the two free ends. These two free ends are connected with one another with the help of smooth muscle called trachealis. Alright, let's look at the histology. I said that trachea contains C-shaped hyaline cartilage present right here. I can say that it's a hyaline cartilage due to the presence of nests of chondrocyte. Now, the free end of the C-shaped hyaline cartilage is connected to smooth muscle, namely trachealis. Now, let's look at the various layers. So, innermost is mucosa. The mucosa contains epithelial mucosa. The epithelial mucosa is none other than respiratory epithelium or pseudostratified ciliated columnar epithelium. Below epithelial mucosa is lamina propria. Then we have submucosa. The submucosa contains lots of glands. These are seromucous glands. Remember, serous glands, they are filled with serum. That is why they are seriously stained. That is, they are stained dark. On the other hand, mucous glands during the preparation of the slide, the mucus gets washed out. As a result, the cells, they appear empty. That is why they are very lightly stained. You can see right here. All the darkly or seriously stained glands are your serous glands, and the lightly stained gland with cells that appear empty are mucous glands. You can see a structure right here. This is seen only during the preparation of slide. This is not something that is seen naturally. And what exactly is this? See, what you can see is a mucous gland with empty cells, right? And it is wrapped from one side with a serous demilune. Serous because it contains serous cells. Demilune because demi means half and lune means moon. So the structure looks like a half moon wrapping the mucous gland from one side. So serous demilune. Alright, then there's your hyaline cartilage with free end connected to trachealis, and outside that's adventitia. Adventitia contains blood vessels. Here's the histological slide. There's your C-shaped hyaline cartilage, the free end of which is connected with trachealis muscle. Here's the C-shaped hyaline cartilage, the free end of which is connected with the help of trachealis muscle. Most students have difficulty distinguishing between intrapulmonary bronchus and trachea. The reason being, trachea also has hyaline cartilage and intrapulmonary bronchus also has hyaline cartilage. But the separating or the distinguishing point is that in trachea, the hyaline cartilage is present as a continuous C-shaped band, and the free end of which is attached with trachealis muscle. However, in intrapulmonary bronchus, the hyaline cartilage is present in parts, and these parts, they are not connected to one another by any smooth muscle. They are not connected. However, an extra layer of muscle is present encircling the mucus layer. The epithelium is still the same, that is the respiratory epithelium or the pseudostratified ciliated columnar epithelium. Here's the histological slide. See, if such a slide is given, it's much easier to identify because There's your alveoli, and then there's your hyaline cartilage. And there's no way that this can be trachea since there's alveoli right here. So it has to be intrapulmonary bronchus. But let's say such a slide has been provided where you have absolutely no idea of presence of any alveoli. So the best distinguishing factor is look at hyaline cartilage, and if they are present in parts not connected to one another by any smooth muscle, it's intrapulmonary bronchus. Next, we have the histology of lung. But before getting into the histology of lung, let's look at two important structures that you can see in the section of lung. That is first, the terminal bronchiole. Before this, we talked about trachea, and then we talked about intrapulmonary bronchus. So we have talked about bronchus. Now we are entering into the world of bronchiole. As soon as you reach bronchiole, there is no longer any hyaline cartilage found. So you can see right here, this is the terminal bronchiole, and the terminal bronchiole does not contain any hyaline cartilage. Hyaline cartilage is found in trachea and bronchus, but not in bronchiole. No hyaline cartilage in bronchiole. Second, so let's look at the layers. You have mucosa, innermost is epithelial mucosa, and the epithelial mucosa now is not a respiratory epithelium. Now it is simply simple columnar epithelium. There are no cilia found, and it is not pseudostratified. It is simple columnar epithelium. Below epithelial mucosa, there is lamina propria, and outside lamina propria, there is a continuous band of smooth muscle. Now, this band of smooth muscle pulls part of lamina propria and mucosa. As a result, these mucosal folds are formed. So, what are the important features? Number one, no hyaline cartilage. No hyaline cartilage in bronchiole. Number two, innermost epithelial mucosa is simple columnar epithelium. Then there's a continuous circular band of smooth muscle which pulls parts of mucosa and lamina propria. As a result, mucosal folds are formed. After terminal bronchiole, you have another structure called the respiratory bronchiole. Respiratory bronchiole also has the lining epithelium and the smooth muscle. However, they are broken. So you will find a discontinuous band of smooth muscle and epithelium. The epithelium present now is columnar to cuboidal. Since the lumen is getting narrower and narrower, you can't adjust columnar cells anymore. So it starts getting cuboidal in shape. So it ranges from columnar to cuboidal, again without any cilia. Obviously, you don't have cartilage because you are in bronchiole, and you don't have cilia because you are in bronchiole. Now, since these discontinuous bands, they connect the space with alveoli. They outreach with alveoli, forming alveolar out pockets. And that's your structures present within the lung. In order to identify alveoli, it has to be the easiest thing ever. I mean, all you have to look for are these sacs, alveolar sacs, and these alveolar sacs, they are lined by simple squamous epithelium. And you have variety of alveolar cells found like pneumocyte 1, pneumocyte 2. You can read them on your own. So this is the histological slide. You can see lots of air pockets or alveolar sacs. So this is obviously the histology of lung or alveoli. All right, one last revision of complete histology slides of respiratory system. First, epiglottis. Epiglottis has an elastic cartilage core which is covered by mucosa. The mucosa contains epithelial mucosa and lamina propria. The epithelial mucosa on the anterior surface and the upper part of posterior surface is lingual mucosa, that is non-keratinized stratified squamous epithelium, and on the lower portion of the posterior surface is laryngeal mucosa, which is pseudo-stratified ciliated columnar epithelium. The submucosa contains two types of glands: serous glands that are seriously or darkly stained, and mucous glands with empty cells that are lightly stained. Second is trachea. Trachea contains a continuous band of C-shaped hyaline cartilage with its free end connected with trachealis muscle. The mucosa contains the same respiratory epithelium, that is the pseudostratified ciliated columnar epithelium. The submucosa contains two types of glands, the seriously stained serous glands and the lightly stained mucous glands. You can also see mucous glands with serous demilunes. After trachea, we have intrapulmonary bronchus. The way to differentiate between trachea and intrapulmonary bronchus is that trachea has a continuous C-shaped hyaline cartilage with its free end connected with trachealis. On the other hand, intrapulmonary bronchus contains hyaline cartilage in parts, and these parts, they are not connected with one another by any smooth muscle. However, there is an extra layer of muscle surrounding the mucosa. Then we have lung with various structures. So we are done with trachea and then bronchus. Now we are entering into the world of bronchiole. So first you have your terminal bronchiole. Since it's a bronchiole, it does not have any hyaline cartilage. And the epithelium is also not respiratory epithelium. It is simple columnar epithelium. Due to the presence of circular muscular fold, the formation of mucosal fold takes place due to circular muscular band. Then we have respiratory bronchiole. The respiratory bronchiole also contains the epithelium along with smooth muscle. However, they are present in parts, that is, there is discontinuous band of smooth muscle and epithelium. As a result, the space inside is in direct communication with alveolar sac, forming alveolar outpockets. The epithelium again is columnar to cuboidal. As for alveoli, they are the easiest to identify. They are present in the form of alveolar sacs that are lined by simple squamous epithelium with type 1 pneumocytes and type 2 pneumocytes.